Hi guys, I am Noura. My colleagues and me from Biotechnology Group F will talk about monoclonal antibodies, abbreviated to MAB. What are they? Monoclonal antibodies known as monospecific antibodies. Mono means one or single. Monospecific antibodies means that they are specifically targeted to a particular antigen or a particular epitope. They bind only to a single particular epitope, so they are specific antibodies.
Well, of course, the beginning types of monoclonal antibodies were not really identical to the human ones, or to the sequences of the human genome. Accordingly, they started to produce monoclonal antibodies from murine type. As you can see here, we have 4 different forms. The first one is murine. Murine, being a word from the Latin word murinus, means mouse. It's derived from the fusion of immortalized mice myeloma cells with the lymphocytes from immunized mice. So here the C region or the constant region diminished the efficacy of the drug, increasing the likelihood of complications. This problem made them make a chimeric monoclonal antibody. It combines both mouse variable region and also a human constant one, and this increased the efficacy greatly. There are another kinds of MABs called humanized, in which the CDR part only is murine. Finally comes to the full human antibody, which is the base type. So you now can see here the difference of human sequences in each type, decreasing the side effects as we go from murine to full human antibody.
Techniques of monoclonal antibodies production. Number 1. Hybridoma, which is the injection of an antigen into an animal, then extracting beta cells that contain the formed antibodies. Fusion between antibodies and cancer cells, which are myeloma, occurs. Finally, implantation of this fusion into tissue culture.
Phage display is one of the techniques used for preparing monoclonal antibodies. First, we will take some antibody genes and combine them on a strand DNA, and then packing it with a protein coat made from virus. Antibody genes are making antibody hat, which binds to the top of the virus coat. Virus coat binds to the phage, which makes thing called the phage antibody hat that can be specific for certain disease. To target a particular disease, we will need to find the right phage antibody. In detail, we are isolating B lymphocytes from blood of human, then isolating the mRNA and converting it into complementary DNA using PCR to amplify VH and VL segments. These segments can be cloned into vector and then used to infect E. coli. The E. coli produce the bacteriophage containing segments which acting against the antigen that can be easily selected to use against certain disease.
Here are some of the applications such as HCV, HIV, and psoriasis. The antiviral strategies are based on interfering with virus infection or spread, as it can block the receptor engagement by binding to specific surface proteins. Some antibodies excrete protective actions through Fc-mediated effector functions, such as antibody-dependent cell-mediated cytotoxicity and complement-dependent cytotoxicity. MBL-HCV1 is a human monoclonal antibody isolated from transgenic mice and directed to HCV E2 glycoprotein, which can neutralize viruses of many HCV genotypes.
HIV can be treated by monoclonal antibodies such as Pro-140 and ipilimumab. Host cell consists of two types of receptors, CCR5 and CD4. Examples of monoclonal antibodies used for treatment of HIV. First, Pro-140 is a monoclonal antibody binds to CCR4 receptor and inhibits the entry of the virus into the cell. Second, ipilimumab is a humanized monoclonal antibody binds to the second domain of CD4 receptor. It prevents viral entry and diffusion to the cell.
Psoriasis is a common chronic skin disorder characterized by cutaneous inflammation, angiogenesis, and keratinocyte hyperproliferation. Antivascular endothelial growth factor antibody can lead to normalization of epidermal architecture and the treatment of psoriasis. Antivascular endothelial growth factor antibody could inhibit induced angiogenesis and improves the skin lesions of psoriasis. One of the examples of monoclonal antibodies against vascular endothelial growth factor is bevacizumab, which is also known as Avastin. It's not only used for treatment of psoriasis, but also for treatment of multiple types of cancer.
Here are some examples of approved monoclonal antibodies for human use. Adalimumab for the treatment of Crohn's disease and juvenile idiopathic arthritis. Golimumab for the treatment of psoriatic arthritis and ankylosing spondylitis. Ustekinumab for the treatment of plaque psoriasis and sarcoidosis.
Future work on monoclonal antibodies. Due to the selectivity of monoclonal antibodies, researchers are working on attaching these antibodies via chemical linkers with drugs needed to be targeted to specific cells, as cytotoxin and chemotherapeutic drugs in cancer treatment. This conjugate's action is directed to specific antigens overexpressed on tumor cells. One of the antibody-drug conjugates that based on humanized monoclonal antibody is called EV20 that recognizes HER3, the human epidermal growth factor receptor 3, coupled to the eukarmycin derivative, a potent anti-tumor antibiotic. This ADC cures tumors overexpressing HER3 as pancreatic cancer and gastric cancer.